Next, vulva induction in Caenorhabditis elegans is a well-studied example of cell-cell communication during development. In C. elegans, the vulva is a multicellular organ that is essential for reproduction. Vulval induction involves the coordinated signaling between two different cell types, the anchor cells and the vulval precursor cells, to generate a functional vulva. So, establishment of the anchor cell. During the L3 stage of C. elegans development, one of the 6 cells in the hermaphrodite gonad, called the AC anchor cell, migrates towards the overlying epidermis. The AC stops at a specific location called the ventral midline and secretes signaling molecules that initiate vulval induction. Signaling from the anchor cell. The AC secretes 2 types of signaling molecules, the epidermal growth factor EGF and Lin3 protein. These signaling molecules bind to receptors on the neighboring cells called vulval precursor cells or VPCs and initiate a series of events that lead to the differentiation of VPCs into vulval cells. Then determination of the VPC fate. In response to the signaling from the AC, the 6 VPCs or vulval precursor cells adopt different fates depending on the position relative to the AC. The VPC closest to the AC becomes the primary vulval precursor cell like P6P, while the 2 VPCs on either side of the P6P cells become secondary vulval precursor cells P5P and P7P. The remaining 3 VPCs adopt a non-vulval fate. Signal amplification and feedback. The signaling from the AC is amplified by a positive feedback loop involving the EGF receptor LET23 and RAS MAPK signaling pathway and the transcription factor Lin1. This feedback loop ensures that the VPCs adopt the correct fate and generate the proper number of vulval cells. Then differentiation of the vulval cells. The VPCs that adopt the vulval fate differentiate into specialized cells including the vulval muscle cells, the vulval epithelial cells and the vulval syncytium. These cells from the mature vulva, which is essential for reproduction in C. elegans. Examples of genetic mutations that affect vulval induction in C. elegans include LET23. A mutation in the EGF receptor gene LET23 results in defective vulval induction as the VPCs fail to respond to the signaling from the AC. Lin3. A mutation in the ligand gene Lin3 results in defective vulval induction as the AC fails to secrete the EGF and Lin3 signaling molecules. Then Lin1. A mutation in the transcription factor gene Lin1 results in abnormal vulval induction as the positive feedback loop is disrupted, leading to the improper differentiation of the VPCs. In summary, vulval induction in C. elegans is a complex and highly regulated process that involves signaling between the anchor cell and the vulval precursor cell, the determination of the VPC fate, the amplification of the signaling by a positive feedback loop, and the differentiation of the vulval cells. This process is essential for the development of functional vulva, which is important for reproduction in C. elegans. Next, Drosophila development in detail. Drosophila melanogaster, commonly known as the fruit fly, is a model organism widely used in developmental biology studies due to its rapid development and genetic tractability. So here is an overview of major steps and factors involved in Drosophila development. Fertilization. After mating, sperm from the male enters the female's ovary and fertilizes the egg, forming a diploid zygote. Then cleavage. The zygote undergoes a series of rapid cell divisions called cleavage, which partitions the cytoplasm into smaller cells called blastomeres. Then formation of blastoderm. After 13 rounds of cleavage, the nuclei migrate to the periphery of the cell, egg cell, forming a single layer of nuclei called the blastoderm. Then segmentation. During the subcellular blastoderm stage, the embryo undergoes a process called segmentation, in which the embryo is divided into distinct segments along the anterior-posterior axis. This process is regulated by genes called segmentation genes, such as the GAP genes, pair rule genes and segment polarity genes. Next, gastrulation. During gastrulation, the embryo undergoes a complex morphogenetic process that results in the formation of 3 germ layers, the endoderm, mesoderm, and ectoderm. This process is regulated by genes such as the dorsal ventral patterning genes, which establish the dorsal ventral axis of the embryo. Then organogenesis. During the later stages of embryonic development, the germ layer differentiate and form specific organs and tissues such as the nerve system, muscles, and gut. This process is regulated by genes such as the homeotic genes, which determine the identity of different body segments. Then examples of genetic mutations that affect Drosophila development include bicoid. A mutation in the bicoid gene, which is required for anterior patterning, results in embryos lacking anterior structures. Then hunchback. A mutation in the hunchback gene, which is required for proper segmentation and anterior patterning, results in embryos with abnormal segmental boundaries and head defects. Then hedgehog. A mutation in the hedgehog gene, which is required for proper dorsal ventral patterning, results in embryos with abnormal dorsal ventral polarity and defects in the nerve system. In summary, Drosophila development is a highly regulated process involving a series of complex morphogenetic events including fertilization, cleavage, segmentation, gastrulation, and organogenesis. This process is regulated by a complex network of genes that determine the identity and fate of the different cells and tissues in the developing embryo. The genetic tractability of Drosophila makes it an excellent model organism for studying the molecular and cellular mechanisms underlying development. Then, introduction and competence in detail. Introduction and competence are two important concepts in developmental biology that play a critical role in the development of tissues and organs. Introduction refers to the process by which one group of cells or tissues influence the fate of another group of cells or tissue. This process involves the secretion of chemical signals by the inducing cells which bind to receptors on the responding cells and trigger a cascade of intracellular signaling events. The inducer cells release signaling molecules. The first step in introduction is the release of signaling molecules by the inducer cell. These molecules can be diffusible factors such as growth factors or morphogens, or they can be membrane-bound ligands that interact with receptors on the surface of corresponding cells. The responding cells receive the signal. Once the signaling molecule reaches the corresponding cells, they bind to specific receptors on the surface of the cells, triggering a cascade of intracellular signaling events. Then transcription factors activate or repress genes. The intracellular signaling events ultimately lead to the activation or repression of specific genes in the responding cells. These genes encode transcription factors that regulate the expression of downstream genes involved in cell fate determination. Then cell fate determination, the activated or repressed genes ultimately determine the fate of the responding cells, directing them to differentiate into specific cell types or tissues. Examples of introduction in developmental biology include the introduction of neural tissue by the notochord in vertebrate embryos, the induction of lens by the optic vesicle in the eye, and the induction of the neural crust by the ectoderm in the vertebrate embryo. Next, competence. Competence refers to the ability of cells or tissues to respond to inducing signals. This process involves the activation of specific signaling pathways and expression of downstream genes that allow the cells to respond to the inducing signals. So, signal reception. The first step in competence is the reception of the inducing signal by the corresponding cells. This can occur through the binding of diffusible factors or the interaction of membrane-bound ligands and receptors. Then signal transduction. Once the signal is received, the corresponding cells activate specific signaling pathways that lead to the expression of downstream genes. Then gene expression. The downstream genes that are expressed in responding cells include transcription factors and other signaling molecules that are necessary for cell fate determination. Cell fate determination. The expressed genes ultimately determine the fate of the responding cells, directing them to differentiate into specific cell types or tissues. So, examples of competence in developmental biology include the ability of neural crust cells to respond to the inducing signals from the ectoderm, and the ability of mesodermal cells to respond to the inducing signals from the notochord in vertebral embryos. In summary, introduction and competence are two important concepts in developmental biology that play a crucial role in the development of tissues and organs. Introduction involves the process by which one group of cells or tissues influence the fate of another group of cells or tissues, while competence involves the ability of cells or tissues to respond to inducing signals. These processes are regulated by a complex network of signaling pathways and downstream genes that determine the fate of developing cells and tissues. Then, Neewoop center. The Neewoop center is a structure located in the anterior margin of the Drosophila wing disc, which plays a critical role in organizing the development of the entire wing. It was first discovered by geneticist Walter Jering in 1968. The Neewoop center is a small cluster of cells that is defined by the expression of several transcription factors, including the homeodomain protein, engrilled EN, and the zinc finger protein, Cubitus interruptus CI. These cells secrete signaling molecules including hedgehog and decapentapelagic DPP, which diffuse across the wing disc and regulate the patterning of the growth of developing wing. The HH signaling pathway is essential for the formation of the anterior-posterior axis of the wing, while the DPP signaling pathway is essential for the formation of the dorsal ventral axis. Together, the signaling pathways specify the identity and growth of different regions of the developing wing. The Neewoop center also plays a role in the regulation of Notch signaling pathway, which is involved in cell fate determination and differentiation. The EN and CI proteins in the Neewoop center represents the expression of Notch target genes, which helps to establish sharp boundaries between different regions of developing wing. In summary, the Neewoop center is a small cluster of cells located in the Drosophila wing disc, which plays a crucial role in organizing the development of the entire wing. It secretes signaling molecules that regulate the patterning and growth of the wing and also helps to establish boundaries between different regions of the wing through the regulation of Notch signaling pathway. Neewoop center in detail. Nape-Koch center is a structure located in the anterior margin of the Drosophila wing disc, which plays a crucial role in organizing the development of the entire wing. The Nape-Koch center was first described by Walter Knape in French Corps in 1922. I'm sorry for mispronouncing these words, so I can't help it. However, it was not until 1968 that geneticist Walter Jering discovered the role of the center in Drosophila wing development. The center is a small cluster of cells that express several transcription factors, including the homeodomain protein, engrilled EN, and the zinc finger protein, Cubitus interruptus. And these cells secrete signaling molecules, including hedgehog and decapentapelagic DPP, which diffuse across the wing disc and regulate the patterning and growth of the developing wing. One important aspect of the center is its role in establishing the anterior-posterior axis of the wing. The HH signaling pathway, which is activated by HH secretion from the center, is essential for the formation of AP axis. HH signaling induces the expression of target genes such as decapentapelagic DPP and wingless in the posterior compartment of the HH signaling induces the expression of target genes such as decapentapelagic and wingless in the posterior compartment of the wing disc, leading to the formation of a concentration gradient of DPP and WG proteins along the AP axis of the wing. This gradient of signaling proteins helps to specify the different regions of the wing, including the notum, hinge, and blade regions. Another important aspect of this center is its role in regulating the expression of Notch target genes. Notch signaling is involved in cell fate determination and differentiation and is regulated by interactions between neighboring cells. The center plays a crucial role in establishing sharp boundaries between different regions of the developing wing through the regulation of Notch pathway. Specifically, the EN and CI proteins in the center represent the expression of Notch target genes. which helps to establish the boundaries between different regions of the developing wing. In terms of advantages and disadvantages, one advantage of the center is that it provides a key organizing center for wing development, ensuring that the different regions of the wing are properly specified and differentiated. However, a disadvantage is that any disruptions to the signaling pathways regulated by this center can result in abnormalities in wing development and growth. So, in conclusion, the center is a crucial organizing center of Drosophila wing development. It secretes signaling molecules that regulate the patterning and growth of wing and also helps to establish boundaries between different regions of the wing through the regulation of notch signaling pathway. Its importance is establishing the apiaxis of the wing and regulating cell fate determination make it a key target for further study in developmental biology. Then fate of germ layers. During embryonic development, the fertilized egg gives rise to three distinct layers of cells known as germ layers, which subsequently differentiate into all of the tissues and organs of the body. These three germ layers are the ectoderm, mesoderm, and endoderm, and each layer gives rise to specific tissues and organs. So, ectoderm. The ectoderm is the outermost layer of cells in the developing embryo. It gives rise to the skin, hair, nails, and lining of the mouth and anus. Which parts? Skin, hair, nails, and the lining of the mouth and anus, ectoderm part. The neural crust, which is a group of cells that forms at the boundary between the neural plate and the surface ectoderm, gives rise to a variety of cell types including sensory neurons, pigment cells, and cranial cartilage. The ectoderm also gives rise to the nervous system including the brain and spinal cord. Then mesoderm. The mesoderm is the middle layer of cells in the developing embryo. It gives rise to the muscles, bones, cartilage, and connective tissues of the body. The mesoderm also gives rise to the cardiovascular system including the heart, blood vessels, and blood cells. In addition, the mesoderm gives rise to the kidneys, gonads, and the lining of the body cavities. Then ectoderm is over. So next, endoderm. The endoderm is the innermost layer of cells in the developing embryo. It gives rise to the lining of the digestive tract as well as respiratory and urinary tracts. The endoderm also gives rise to the liver, pancreas, and thyroid glands. The importance of germ layer formation lies in the fact that it is the initial step in the development of an entire organism. The formation of the germ layer sets the stage for the differentiation and specialization of cell types that will occur during later stages of embryonic development. Additionally, defects in germ layer formation or differentiation can lead to developmental abnormalities and birth defects. For example, a defect in ectoderm formation can result in neural tube defects such as spina bifida or anencephaly as well as skin and hair abnormalities. A defect in mesoderm formation can result in skeletal abnormalities such as scoliosis or cardiovascular abnormalities such as congenital heart defects. A defect in endoderm formation can result in digestive tract abnormalities such as esophageal atresia or in respiratory abnormalities such as pulmonary hyperplasia. In summary, the fate of each germ layer is critical to the development of an organism. The defects in germ layer formation or differentiation can lead to significant developmental abnormalities. Understanding the differentiation pathways and signaling mechanisms that regulate germ layer formation is therefore a key focus of research in developmental biology. Then morphogenetic movement. Morphogenetic movements are a series of coordinated cellular processes that occur during embryonic development, leading to the shaping and patterning of body plan. There are several types of morphogenetic movements that occur during development, including convergent extension, invagination, involution, and epiboly. So, convergent extension. Convergent extension is a process that occurs during gastrulation in which cells in the dorsal mesoderm and endoderm migrate towards the midline of the embryo, leading to the elongation of the body axis. This movement is mediated by the contraction of the actin and myosin filaments within the cells, leading to the intercalation of cells and the narrowing of the tissue along one axis while elongating it along the perpendicular axis. Then invagination. Invagination is a process that occurs during gastrulation in which cells in the outer layer of the embryo fold inward, forming a pocket of indentation. This process is seen in the formation of archenteron during gastrulation in many animals including sea urchins, frogs, and chickens. Then involution. Involution is a process that occurs during gastrulation in which cells on the surface of the embryo move inward, rolling over and migrating to the interior side of the embryo. This process is seen in the formation of mesoderm and endoderm in many animals, including frogs and sea urchins. Then epiboly. Epiboly is a process that occurs during gastrulation in which cells from different regions of the embryo migrate and spread over the surface of the yolk, leading to the expansion of the embryo. This process is seen in the formation of the ectoderm in many animals, including zebrafish and frogs. The importance of morphogenetic movements in embryonic development lies in their role in shaping and patterning the body plan. The coordinated movements of cells during development leads to the formation of complex three-dimensional structures, including the brain, heart, and limbs. Additionally, defects in morphogenetic movement can lead to developmental abnormalities and birth defects. For example, defects in convergent extension can lead to neural tube defects and skeletal abnormalities such as spina bifida or scoliosis. Defects in imagination can lead to abnormalities in the formation of achondron, which can result in digestive tract abnormalities. Defects in involution can lead to abnormalities in the formation of mesoderm and endoderm, which can result in cardiovascular or digestive tract abnormalities. In summary, morphogenetic movements are critical processes in embryonic development that are essential for shaping and patterning the body plan. Understanding the cellular mechanisms and signaling pathways that regulate these movements is therefore a key focus of research in developmental biology, as defects in these processes can lead to significant developmental abnormalities. Next, genetics. So, Mendelian inheritance in detail. Mendelian inheritance, also known as classical genetics, is the basic principle of inheritance governed by Gregor Mendel in the mid-19th century. It describes the way traits are passed down from parents to offsprings through the transmission of genes. There are three major principles of Mendelian inheritance: the law of segregation, the law of independent assortment, and the law of dominance. So, law of segregation. This law states that pairs of genes, which are alleles, separate during the formation of gametes so that each gamete receives one allele for each trait. This is the basis of Punnet's square method of predicting the probability of offspring inheriting a particular trait from their parents. Then, law of independent assortment. This law states that the inheritance of one trait is independent of the inheritance of another trait, meaning that genes for different traits are sorted into gametes randomly and independently of each other. This principle applies only to genes located on different chromosomes or genes that are located far apart on the same chromosome. Then, law of dominance. This law states that in a pair of alleles, one allele is dominant and another one is recessive. This dominant allele will be expressed in the phenotype, while the recessive allele will be masked in the presence of the dominant allele. So, an example of Mendelian inheritance is the inheritance of eye color in humans. The gene for eye color has two alleles, brown and blue. The brown allele is dominant and the blue allele is recessive. If an individual inherits one brown allele and one blue allele, their eye color will be brown because the brown allele is dominant. However, if an individual inherits two blue alleles, their eye color will be blue because the blue allele is recessive. The importance of Mendelian inheritance lies in its fundamental role in genetics research and understanding inheritance patterns in many organisms. By understanding the basic principles of Mendelian inheritance, scientists can predict the likelihood of certain traits being expressed in future generations and develop new techniques for genetic engineering and selective breeding. Additionally, Mendelian inheritance has important implications for human health, as many genetic diseases are caused by mutations in single genes that follow Mendelian inheritance patterns. In summary, Mendelian inheritance describes the basic principles of how traits are passed down from parents to offspring during the transmission of genes. Understanding the laws of segregation, independent assortment, and dominance is important for predicting the likelihood of certain traits being expressed in future generations and developing new techniques for genetic engineering and selective breeding. Next, 3-point mapping. Three-point mapping is a method of genetic analysis used to determine the relative locations of three genes on a chromosome. It was developed by Alfred Stuart Wand. I'm not sure about the pronunciation. Stuart Wand in the early 20th century as an extension of his two-point mapping technique, which allowed for the determination of the relative location of two genes on a chromosome. Three-point mapping is an important tool for genetics in understanding how genes are arranged in chromosomes and how they are inherited. The three-point mapping technique involves crossing two individuals that are heterozygous for three linked genes. The frequency of the different offspring genotypes is analyzed to determine the order and distance between the genes. The following are the major steps involved in three-point mapping. Crossing over. During meiosis, homologous chromosomes can exchange genetic material through a process called crossing over. This results in new combinations of alleles on the chromosomes. Then analysis of offspring. The offspring of the heterozygous parents are analyzed to determine the frequency of each of the different genotypes. Then calculation of recombination frequencies. The recombination frequency between two genes is the proportion of offspring that have recombinant genotypes. In three-point mapping, the recombination frequency between the two outermost genes is calculated first. So calculation of recombination frequencies. First step was crossing over, then analysis of offspring. Next, calculation of recombination frequencies. The recombination frequency between two genes is the proportion of offspring that have recombinant genotypes. In three-point mapping, the recombination frequency between the two outermost genes is calculated first. Then the recombination frequency between the middle gene and each of the outermost genes is calculated. The importance of three-point mapping lies in its ability to provide information about the order and distance between three genes on a chromosome. This information is critical for understanding the mechanisms and inheritance and how genes are arranged on chromosomes. Three-point mapping also allows geneticists to identify regions of chromosomes that are likely to contain multiple genes, which can be important for identifying genetic disorders. An example of three-point mapping is the mapping of genes for eye color, wing shape, and body color in fruit flies. By crossing flies that are heterozygous for these three genes, Stuart was able to determine the relative order and distance between them. He found that the genes for eye color and wing shape were relatively close together on the chromosome, while the gene for body color was further away. In summary, three-point mapping is a genetic analysis technique used to determine the relative order and distance between three genes on a chromosome. It involves analyzing the frequency of different offspring genotypes and calculating recombination frequencies. Three-point mapping is important for understanding the mechanisms of inheritance and identifying genetic disorders. An example of three-point mapping is the mapping of genes for eye color, wing shape, and body color in fruit flies. Three-point mapping in detail with examples. Next, we'll be learning that topic once again tomorrow. Thank you for listening.